Hello everyone, I am Nivedita China from Ambika Prasad Research Foundation. Welcome to all of you to our channel Biodiversity and Conservation. So here today we are going to learn about the microbodies in a cell. So let's start. So what are microbodies? Microbodies are different type of bodies present in the cytosol, also known as cytosomes. A microbody is usually a vesicle with a spherical shape ranging from 0.2 to 1.5 micrometers in diameter, and these are found in the cytoplasm of a cell, but they are only visible with the use of an electron microscope. And these are the eukaryotic cell organelles that are bound by a single membrane. These are absent in prokaryotic cells and are filled with different types of oxidative enzymes, and their main functions involve detoxification of peroxides, oxidative metabolism, and photorespiration in plants. So then coming to the types of microbodies, the major types of microbodies found in higher eukaryotes are peroxisomes, the second one is perosomes, and the third one is glyoxysomes. Apart from these, some protozoans or trypanosomes have glycosomes, which are microbodies containing glycolytic enzymes, and some anaerobic ciliates, flagellates, and fungi have hydrogenosomes, which are microbodies that help in the production of ATP and molecular hydrogen in anaerobic conditions. So the microbodies are categorized into three subcategories is peroxisomes, perosomes, and glycosomes. Peroxisomes is the breakdown of very long chain fatty acids, and it involved in peroxide metabolism in animals and involved in respiration in plants. Sperosomes are the synthesis and storage of lipids, and glycosomes converts fats into carbohydrates. Peroxisomes are the microbodies that are involved in the synthesis of hydrogen peroxide during their metabolism. These organelles were named peroxisomes by D. Dubé in 1969 and were first seen in rat kidney and liver cells with the help of fractionation technique. So the location of peroxisomes in both plant and animal cells contain peroxisomes in their cytoplasm, lying close to the mitochondria, endoplasmic reticulum, and chloroplast in plant cell. And these are derived from the endoplasmic reticulum and are present in numbers of about 7 to 100 in photosynthetic cells. And here the structural organization of peroxisome. The average diameter of peroxisome ranges from around 0.5 to 1 micron. They are bound by a single phospholipid bilayer membrane enclosing a granular matrix and a central core known as a nucleoid, which is crystalline or fibrous in nature, is present in the matrix, and multiple oxidative enzymes such as catalase, peroxidase, uric acid oxidase, D-amino acid oxidase, B-hydroxy acid oxidase, D-amino acid oxidase, etc. are found in peroxisomes. So coming to the functions, peroxisomes help in oxidative detoxification of toxic substances such as phenols, nitrites, formaldehyde, methanol, formic acid, etc., and 25% of the consumed alcohol. They also help in the breakdown of xenobiotics, which cannot be metabolized by other digestive enzymes in the body. Peroxisomes are responsible for metabolism of long-chain fatty acids in animal cells. In photosynthetic plant cells, peroxisomes help in photorespiration. They pick up the glycolate generated in the chloroplast and oxidize it to form glyoxylate, thereby releasing hydrogen peroxide as a byproduct. The glyoxylate is eventually converted to amino acid glycine. And these peroxisomes also help in conversion of nitrogen fixed in the root nodules to acyl derivatives of area or urites, catalysis of condensation reactions, and the removal of amine groups from amino acids by oxidation. Then the last one is glyoxygenes. These microbodies have similarities to the peroxisomes as they contain similar enzymes in addition to the enzymes of the glyoxylate cycle, and they were first explained in 1967 by R.W. Bredenbach. And these glyoxygenes are seen in the cytoplasm of plant cells, germinating seeds of watermelon, peanuts, castor, etc. generally show the presence of numerous glyoxygenes. The structural organization are bound by a single membrane, and their matrix consists of a crystalloid core as seen in figure, and these organelles mostly contain enzymes such as malate synthetase, isocitrate lyase, and many enzymes of the Krebs cycle. And uh, the function of glycosomes help in synthesizing carbohydrates from the lipids present in the endosperm of germinating seeds through the glyoxylate cycle. They help in the oxidation of fatty acids and produce acetyl coenzyme A in the process. This acetyl coenzyme is further metabolized through a glyoxylate cycle to form carbohydrates. Once the stored lipids in the plant cells are mobilized by the glyoxylate cycle in glyoxygenes, they stop acting as glyoxygenes and become peroxisomes. Then they reappear in senescent plant tissues to help in degradation and mobilization of lipids. Then coming to the spirogymes, these microbodies are ovoid or spherical in shape and help in lipid and protein metabolism. Werner discovered spirogymes in 1953. The, its location is the plant cells mainly involved with the storage of lipids such as cells of endosperm tissue of all seeds. So the presence of spirogymes which arise from the endoplasmic reticulum, and the structural organization are bound by a phospholipid monolayer which forms a half unit membrane. The hydrophilic polar heads of the phospholipids are arranged towards the outer side, while the hydrophobic nonpolar tails are arranged to face the inner side. And 98% of the composition of spirogymes consist of lipids, while the remaining 2% consist of proteins. Then the function of spirojum describe that spirogymes of some tissue like tobacco, endosperm, maize, root tip, etc. contain hydrolytic enzymes. Therefore, they are also considered to have lysosomal activity. And the spirosomes help in the storage and synthesis of lipids. A limited variety of hydrolytic activity, similar to the one shown by lysosomes, is shown by the spirosomes of endosperm tissue of tobacco and root tip cells of maize. This is because their spirosomes contain hydrolytic enzymes, and um, uh, plant lysosomes are a common name for spirogymes, and the function in fat synthesis and storage. They can only be found in the cells of plants, and they mostly present in cells that are involved in fat synthesis and storage, as the endosperm of oily cells.